This is Nick Gant from the University of Auckland. I'm here with Clare Turner from my lab, and we'll talk you through this paper. It's clear that sensory signalling in the mouth is much more complex than our rudimentary understanding of the gustatory system, and it's becoming evident that receptors in the mouth encode information about the qualities of food that go far beyond the perception of taste. A compelling example that shows an unknown mode of signalling in action has been seen during physical exercise. In laboratory studies, athletic performance has improved when carbohydrate solutions were merely rinsed in the mouth. These solutions are not swallowed, so can be of no energetic use to the muscle, and performance is not enhanced in this way when taste-matched, energy-free solutions are rinsed. In our previous paper, we linked for the first time these enhancements in behavioural performance with neurophysiological changes in the motor system. By using non-invasive brain stimulation during prolonged arm exercise, we showed that when carbohydrate was in the mouth, excitability of the descending pathway increased, and the force that could be generated by the elbow flexors at that time increased by around 3%. These are dramatic changes for such a subtle and blind nutritional intervention, but what's even more remarkable is this effect was immediate. For this new study, we wanted to use brain imaging to investigate cortical regions involved in the phenomenon. This technique allows us to isolate the activation network associated with a motor task and then interleave this exercise with repeated exposures to carbohydrate and placebo solutions. The apparatus involved an assembly of automated syringe drivers. The taste-matched test solutions, as well as an artificial saliva, were delivered at program rates via individualized mouthpieces. The infusion and suction tubes were positioned so that the liquid was rinsed over the tongue continuously throughout the protocol. The physical activity was hand grip exercise, where force production was maintained using a handheld force transducer. Then visual cues, presented as shapes, indicated periods when force production or a rest condition were required. These different task and solution conditions were arranged in 25 blocks during a 10-minute echoplanar imaging sequence that was completed by 10 participants.
Mouth rinse in a carbohydrate solution increased activation within the sensory motor network that's responsible for the hand movement. This spread of activation, shown by the colour gradients here, was around a third greater when carbohydrate was rinsed compared to the change we saw with the placebo solution. This extraneural activity recorded when carbohydrate was present is task-specific. In other words, it was only detected during physical activity, not when resting. Carbohydrate rinsing also primed cortical regions involved in visual perception during the hand grip task. Given that the visual cues presented at the time of this comparison were identical, the cross symbol, this suggests that carbohydrate enhances early processing of visual information. So a tasteless carbohydrate solution can rapidly alter brain function specific to a motor task. This shows that networks controlling movement are able to rapidly integrate information concerning muscular work with signalling from the mouth concerning forthcoming nutrients.
Taken with the findings of our previous study, the work highlights a unique form of nutrient signalling that is distinct from perceived taste or smell and can immediately increase voluntary force production in skeletal muscle. With this paper, we also introduce a novel experimental platform that is capable of presenting solutions for multiple exposures without ingestion and can detect the resulting neural and behavioural outcomes. Thanks for your interest in our research. We hope you enjoy reading this article.